Hi. If you compare your integrate system with the CRISPR-Cas for specific integrations, so do you think which one is better? And because also you can integrate whatever you want by homologous recombination, using the homologous recombination system. And the second question is if you can integrate with your integrate system the CRISPR-Cas to work together in order to have a more versatile system.
Okay, very good comment. Yes, certainly we are using the CRISPR system and to edit the genome. Our preliminary experiments we did without using the CRISPR or the Cas9 or the Cas9-like enzymes to actually do that. That we are not going to use the Cas9, but similar to Cas9 to edit the genome, but exactly. And you put the right point. We're going to use editing genome and to add integrase as a plus for the addition of the genome, because we are going to localize the exact point within the genome that we want to integrate, and we are going to put both attachments to actually make the gene function in a specific place. Yes, very good comment.
Yes, in your study of the silk, the protein, the spider silk, the mechanical properties are largely, you think, dominated by the intermolecular interactions of the fiber, or it is the property of the individual fiber. In your simulations, you seem to imply that it's the individual molecule that has the mechanical property that dominates. But obviously, in the experiment that you show, you are pulling a macroscopic fiber that is made up of many, many proteins. So which one is it?
Yes, it's a very good question. I didn't have the time to actually put these details, but indeed, they are, in fact, we synthesize modules that we want, and we put repeating modules together. And that is one point that very interesting that you've made, is that we need to have a fiber, we need a protein above 50 kilodaltons. If you produce a protein, synthetic one, with not enough modules, for example, 8 modules, you are going to have a protein with the same characteristics, but you are not going to be able to extrude, as we call it, and transform in fiber. So we have to have a minimum size to actually produce a fiber. We know that in natural, spiders produce proteins up to 300 kilodaltons. We can produce in bacteria about 95 kilodaltons. It is enough for we to produce fibers.
Yeah, but the question I was asking more specifically was whether the mechanical properties that you measure in the fibers are dominated by the intermolecular contacts between the polypeptides, or by the individual polypeptide.
Oh, no, by the individual, yes. They are simply beta sheet, which is the formation and the folding of the protein is a very simple one.
Yeah, no, but I like to suggest that that it would be interesting to compare the mechanical properties of the fiber with the mechanical properties of the individual molecules to see how much of it is contributed.
Indeed, very much so, yeah.
You have time for the last short question, please, Ramon, sorry.
Yeah, in the case of the HIV drug, I have 2 questions. How you find it, and what is the mechanism of action of the drug?
Sorry, the cyanovirine, you mean the HIV, the protein, which was from an algae that I've mentioned, yes. This was out of a project prospecting algae in the ocean. And out of several algae, were found some, not only cyanovirine, but griftesin is another one, cytovirine also. There are 3 or 4 which you have been able to. The interesting feature of cyanovirine is that does not bind to GP120 in only one site, bind in 3 manod sites. So if you bind in 3 different sites, you can imagine that's more difficult to generate tolerant resistance against the drug. This is a very unique and interesting characteristic of the cyanovirine.
Oh, I see.
Thank you.